Hi, in this video I'm gonna explain about in vitro amplification of DNA, or otherwise called as PCR. Before I go into the specifics, let us get to know about the scientist who invented PCR. Carey Mullis in the year 1983 was awarded the Nobel Prize for his invention of PCR. I've added a few more points on PCR and all the awards that Carey Mullis has got in his life. I'm gonna give you a few more seconds to look at the information projected on the screen, or if required you can pause and then read the entire information on the display.
Polymerase chain reaction, or PCR, is an alternative to cloning for isolating a particular DNA sequence. PCR can selectively amplify a single copy of the desired sequence that is present in a complex mixture of other DNA molecules. In this example, a mixture of genomic DNA molecule is shown as blue ribbons, which contains the sequence which we wish to amplify, which is called as a target sequence, which is shown in green. With PCR, we can make billions of copies of the target sequence without having to purify beforehand. In the first cycle of the PCR, the DNA molecules are melted by raising the temperature to 95 degrees. After the strands have separated, the temperature is lowered to 60 degrees so that a pair of short oligonucleotide primers specific for the 3' end of the target sequence can bind, one primer on each strand. The primers then direct a DNA polymerase called Taq to synthesize the complementary DNA strands from free nucleotides. In the entire mixture of genomic DNA molecule, only DNA containing the target sequence is copied because the Taq polymerase can only copy molecules that have a primer attached. At the end of cycle 1, both strands of the genomic DNA have been copied to form two partially double-stranded DNA molecules. In the next cycle, the same steps are repeated. DNA duplexes are once again melted by heating to 95 degrees. Since Taq polymerase has the special property of thermostability, it is not inactivated by temperature. The primer anneals to the target when the temperature is lowered, and the target strands are extended by Taq polymerase. The temperature can then be raised to 72 to speed up the reaction. At the end of cycle 2, 4 partially double-stranded DNA molecules are produced. All contain the target sequence plus some adjacent DNA. During the third cycle, the DNA strands are again melted, the primers hybridize to the target sequence, and the Taq polymerase synthesizes complementary DNA strands. Cycle 3 produces 2 double-stranded molecules that contain only the target sequence, as well as 6 longer molecules that contain the target and flanking sequences. In the subsequent cycles, the number of target sequence molecules will increase exponentially. At cycle 4, the DNA strands are melted, the primers anneal, and the DNA strands are copied as in the previous cycles. 8 molecules of the target sequence and 8 longer molecules are produced. In cycle 5, the heating, annealing, and synthesis steps are repeated. By the end of cycle 5, there are 22 target molecules and 10 longer molecules. The number of target molecules sequences continue to grow exponentially at each cycle. By the end of the 30th cycle, the target sequence has grown to more than 1 billion double-stranded DNA target molecules, whereas there are only 60 copies of the longer DNA molecule. This means that the final product is virtually pure target sequence. Thus, like cloning, PCR selectively generates a large number of copies of the desired DNA sequence from the original mixture of DNA sequence. Thank you for listening to this topic. We'll meet you once again in the next